Thanks, Christina. And thank you all for coming. So the title of my talk is From New Zealand to Zealand, a year-long odyssey of cultural immersion and community ecology. It's a pretty vague title, and I'll explain why. But obviously, New Zealand has to do with this country here. And Zealand is actually this island in Denmark. But that's not where New Zealand got its name from. It's actually named after this region of Netherlands, and that's because of Abel Tasman, who sailed around Oceania, and that's why the Tasman Sea and Tasmania are so named. So, due to the nature of my funding, I have two very distinct projects. In the first part, I'll be talking about my experience in the NSF EPSI program working on a Quarry dieback disease. And then in the second part, I'll talk about my participation in the NSF Grow Program in Denmark, where we're using a metabarcoding study to look at wood decay over time. First into the NSF EPSI. So this is a program that's since now canceled, but it allowed any graduate students in the STEM fields to apply, and 15 of us went to New Zealand, and we had an orientation there, and then sort of went our own ways. So I was working with Maj Padamsi, who's actually a gopher, and she did her research. It was with Dr. McLaughlin, and I believe Peter Kennedy has taken that position now. I was also working with Stan Bellegarde and had help from postdoc Chantel Probst. So I was working with land care research, and like I mentioned, we had a 3-day orientation in Wellington, but then I went to Auckland, the Auckland Center for Land Care Research. And now this project, I only had 8 weeks to complete, so I had very little room for error. The applications are based on how feasible they are to complete within the 8 weeks. So we've since expanded on the project a bit, but what I'll touch on is mostly what was accomplished during those 8 weeks. So first, the problem here. This is a quarry tree, or Agathis australis. They're from an ancient lineage, the Oricariaceae, which diverged about 200 million years ago during the Jurassic. They have distinct life stages where they have this, typical conifer form, but then they shed their branches as they grow and develop into these massive trees. So they are long-living, thought to maybe live up to 2,500 years old, and they're massive. And so I've actually put a couple pieces of tape in here over by Corey and Dr. Blanchette. There are 2 pieces of tape there that show from here to those pieces of tape are the diameters of the 2 largest living species. And just behind Jim and Linda there's another piece of tape. And this is the diameter of the largest reported quarry tree ever. So we could basically have this whole seminar inside of a quarry tree. It's pretty amazing. Quarry forests are the most biodiverse in New Zealand. They're economically relevant, which didn't help them out too much, and they are very culturally important. So before human arrival, just about 900 years ago, compare that to when humans arrived to Europe and North America, there were over 1.2 million hectares of these trees that occupied above the 38th parallel here in northern New Zealand. And then after Europeans arrived, they logged them down between the 1850s and 1950s, or the mid-1900s, to just 7,500 hectares. So really devastating. And then, as a part of reforestation efforts, after it was made illegal to mill the trees, and probably through the nurseries, they introduced this exotic pathogen that causes quarry dieback. So this is caused by Phytophthora agathodicida. It has only one mating type found there, suggesting that it's an exotic pathogen, and it's homothallic, so it can still produce oospores, even though only that one mating type is there. It causes a collar rot disease, and that leads to this heavy gummosis and then defoliation, which ultimately leads to death. Now, control is challenging, and what I'm showing here are the surface roots that grow from the quarry trees. They also have deeper roots, but these surface roots, as we see here, they're really easy to get trampled on and for the pathogen to infect the roots. And they're making a big effort to prevent this disease, prevent further spread of the disease. It's kind of a losing battle right now, so we're looking for more answers to manage the disease. A bit of the context. So this particular tree that I had shown you earlier is a literal deity to the indigenous Maori people, and the most important one, giving rise to all forest animals and directly to humans in their belief system. And as a part of this project, there's a large cultural component. So we're taking into account the Maori conservation principles when thinking about how to manage this disease. So in general, we can think of conservation in Western science as backwards looking, where there's been some sort of disturbance and it's changed something, and our conservation efforts are generally aimed at getting back to some point, and that we typically think of ourselves as separate from nature. And that stands in opposition to Maori practices, where they have a forward-looking view and think of conservation as more of a game management strategy, where because of the deep connections they've had with forest resources and the land, that they see the land as being responsible for taking care of them and them taking care of the land. Now, of course, this is treading on thin ice here, because it's not a monolith, right? This is a living, Maori people are still there. Many of the kids care more about hip hop than trees, right? But in terms of this disease, the leaders that people from land care research have talked to say that now the quarry tree is on a path forward with this disease, and that we need to find a way for them to coexist with, so it's not something where eradication in large amounts of chemical control will be accepted. So Maj and others were looking at the arbuscular mycorrhizal associations with quarry trees, and found that there are about between 35 and 60 species associating with them. And during that time, they were looking at microscope images and saw that there were fungi in there that had septa. Of course, arbuscular mycorrhizal fungi don't have septa, so they were wondering, are these dark septate endophytes? Now, dark septate endophytes is kind of a broad term. It's a polyphyletic lifestyle or morphology based group, and in the class, they're class 4 endophytes, these non-clavicipitaceous endophytes. They're fairly understudied, so just doing a Web of Science search, we get over almost 24,000 hits for mycorrhizae or mycorrhizal, but only 437 for dark septate endophyte. They're mostly ascomycetes, and usually from Holochiales. And like we know about mycorrhizae that Nancy Johnson was telling us about, these things exist along this mutualism parasitism continuum. So when this idea was proposed by Maj to look at the dark septate endophytes, I went into the literature and found that there were some studies looking at the effect of dark septate endophytes inhibiting the growth of other Phytophthora pathogens. And one example here is this pathogen, Phytophthora citricola. And here we're looking at different dark septate endophytes in a control inhibiting the growth of the pathogen in pure culture. So they saw that this Phylocephala dark septate endophyte was inhibiting the pathogen quite a bit. They extracted the secondary metabolites and found these secondary metabolites, characterized them, and tested them individually against the pathogen, and showed that it was also antagonistic. So this just led me to the obvious question, are any of the quarry-associated dark septate endophytes antagonistic to Phytophthora agathodicida? So we took a culture-based approach, and now I've talked about culture here, but what I mean by culture-based is that these are fungi that can be grown in culture. Some, like arbuscular mycorrhizae, other fungi are not culturable. So we were up here in Auckland. We had 2 field sites. We only collected roots from asymptomatic trees, went back, surface sterilized those, plated them on a few different media, and then the ones that grew out, we got in pure culture and sequenced the ITS. Then put the ones that grew out fast enough to screen through plate inhibition assays. And in these, putting the dark septate endophyte pitted against the PTA, we would expect that as they grow, that if it was antagonistic, this Phytophthora would stop growing, avoid the endophyte. And that if we just had a mock plug there, it would keep on growing over it. And then here we used 2 levels of positive control, and this is the same chemical that is sprayed on boots, so it's known to kill Phytophthora. And then I just looked at sort of these margins here under the microscope to see what the mycelium looked like. So some of the early results here. So from 309 plates, 217 isolates were sequenced. Here we're looking at the number of OTUs and the isolates per cluster. So there were 49 of one isolate that is one species here. And then we have a pretty typical distribution where most species are rare and some are very common. So there were 57 species at the 97% clustering level. I plated both linear root pieces and the nodules alone. We see that 38 only came out from the linear, 10 from both, and 9 from the nodules. Now, from here, we're looking at the phylum or subphylum level on the outside. We can see that Ascomycota dominate in the isolate and in the OTUs. And that 13 of the orders, there are 13 orders represented, 11 of those are Ascomycota, and that the Helotiales dominates, especially looking at the isolate level, but at the OTU level also, and then followed by Hypocreales, Eurotiales, and then Sorderiales are also fairly common. Of these, 13 of them didn't match at 90, so there was a less than 95% match in all blast hits, suggesting that 13 of these were new species that would need to be described. Okay, so this plate inhibition assay, I mentioned that these were our controls, and to show you that they work, here's day 0, day 4, day 7, and day 13. We see that's fully grown over. We have more inhibition here than there. So of the 34 species that were screened, 11 of them strongly inhibited Phytophthora. 22 grew out too slowly or have since been isolated that haven't been screened, and I'm working on getting those here. And here are 2 examples. So this Cryptosporidiosis or Posicula and a Leotiomycetes, both of these are fairly common dark septate endophytes. Here's day 0, day 4, day 7, and day 13. Now, if we look at in this margin of the control mycelium of Phytophthora, we see that in a negative control, this is what regular mycelium looks like. In a positive control, this was the 5%. We see this sort of bulging, swelling of the mycelium, and it's common for hyperbranching to occur. So looking at the margin here, on this Cryptosporidiosis, we see this massive hyperbranching and bulging, and also at this Leotiomycetes where this pigment is, where there's the same sort of effect. And so that leads me to wonder what these secondary metabolites are, and we hope to do some work on that. So some future directions: we've got to screen the other 22 dark septate endophytes, use bioguided fractionation to get at the secondary metabolites, characterize any secondary metabolites, and test the pure metabolites against Phytophthora. Then, since I've been gone, they've hired some other summer students. They're looking at the differences between symptomatic and asymptomatic root communities. What we can do is do math, and hopefully we come up with this Bray-Curtis dissimilarity that we can plot on NMDS plots. Then finally, down the line, I hope to combine sort of what we learned from these inhibition assays the differences between the communities to see what's in healthy communities, and put these, align this with the Maori conservation principles to select some of these endophytes and find a way to deliver them in the biocontrol method back to the quarry roots to help prevent future infections so that we can keep quarries standing. Okay, now we're gonna totally switch gears. This is my Men in Black mind wipe so you forget everything. I'm gonna move on to part 2. Okay, so here in the NSF Grow Program. Now, the Grow Program is still ongoing, but it's only available if you have this GRFP funding, which means that the competition was a bit less for this fund. It was nice. I found a host and developed a project with Jakob Hilman Clausen from Denmark, and he's at the University of Copenhagen and the Center for Macroecology, Evolution and Climate, or SMEC. So here's Jakob, and he's been thinking a lot about fungal diversity in wood and conservation of diversity, and lately he's been working on these networks, interaction networks. So why do we care about wood? Well, it's an incredibly abundant resource that's full of cellulose. It's the most abundant biopolymer, lignin, which is the second or third most abundant, and hemicellulose. So together, this is this massive carbon pool, and the better that we can understand the rate and fate of these products, we can more accurately model carbon dynamics, and yeah, I guess that's important. Also, biodiversity there's a lot of biodiversity in wood that's not often thought of. So many fungi, bacteria, and micro invertebrate animals. And we know that although this paper has received some recent criticism, which I don't think was totally fair, we know that biodiversity is important. We have to maintain biodiversity for all the ecosystem services that are provided, and also sort of the intrinsic and relational values that we have. Now finally, I think that wood is a really cool study system, partly because we have this very heterogeneous matrix. The composition changes between different species. The chemicals that are in there change between different species. And we have, over time, this very finite cycle, as opposed to something like studying a temporal study in soil, where it's more of a continuum. And there are many, when we look at how wood communities develop, wood-associated communities develop, they're very heterogeneous as well. So we have many interactions going on. If each of these was sort of a community in there, or dominant fungus or other organism. So it's important for looking at nutrient cycling, asking many trait or function-based questions. So I want to go back to 1983 and think about how people were viewing the decomposition of dead wood. So it was thought dead wood was sort of studied as this material that was sterile and only became habitable after some disturbance event. Now, I should note that it was known that there are wood decaying pathogens, but they were generally studied in isolation. So after this material became available, it was thought there was this neat succession where these rutteral or stress-selected organisms came in and were fairly ephemeral. They would be supplanted by airborne, combative selected decayers, and later, as the competition increased, the strongest combatants would take over these soil-borne, cord-forming, aggressive decayers. Now, it was thought that this niche sort of became more specialized, and that as decay progressed, richness would go down. Recently, that's been challenged by molecular work, where a lot of this was, of course, morphology-based. And so molecular work is showing, at least in some studies, and this study is from a Norway spruce forest, that richness actually increases as decay progresses. And I want to stress that this is not due to naivete, right? There was very careful studies done tracking decay progression over time, but just limited by the technology. So around the same time, the idea of endophytes was becoming popular, and especially led by George Carroll, who found these internal fungi in old growth fir forest, and so foliage, excuse me. So sort of the confluence of these ideas was that perhaps there are these what are now known as latent saprotrophs existing in the wood as endophytes that at some point would be triggered. And there were a couple of lines of evidence, and a lot of this was brought about by Lynn Boddy, and who later went on to write, be a part of this paper of, do all trees carry the seeds of their own destruction? And it seems like yes. And so a couple things that she recognized was that looking at the maximum linear growth rates in laboratory studies of wood decaying organisms, it didn't match up with a point infection in the rapid decay that would be observed in nature. So it also was shown that some of these fungi could grow in 100% CO2 environments, and that moisture content could suppress growth. And that was shown by using progressive drying regimes and seeing when decay would happen. So she said, what if there are these modules or these propagules that are present extensively but aren't active until something triggers them to be active, and at that time they all sort of give rise to this rapid decay, and that we shouldn't conflate sort of the dominance of any fungus there with any arrival time. So that brings us to this idea of priority effects. Why would that even matter? So a lot of this work is led by Tad Fukami, who happened to be visiting SMEC on sabbatical when I was there, and who had previously visited Auckland Land Care, which is pretty odd. And a lot of his work has been to show that if you have some species pool and there are these niches that are available, if one particular organism gets there, it will change what can arrive afterwards until there are these filled niches. Another way this can happen, other than just preemption, is by modification. So here we have a species pool. There are some initial niches, and depending on what arrives there and what it modifies will dictate what can fill in those spots later. And there are three ways that it's thought that this can occur. One is through the mainland island model, where we have a species pool in these various local communities. Another is a meta-community model, which may be more likely to happen in the lab if we brought something in and inoculated it. But what we're working with here is the integrated model, where the forest and everything in it would be the species pool, and then each log or part of a log would be this sort of isolated activity. So does this actually happen? Well, Tad Fukami and others looked at Southern Hemisphere beech stem rounds and sterilized these. They changed the order that the first fungus was arrived, and then later added different fungi. And they showed that this affected, and then they cut this piece up and looked at what species were there, the percent colonization, and how much CO2 would be released. And they found that all of these things were affected by which fungus arrived first. Okay, but this is all in dead wood that was sterilized. So more recently, in Jonathan Schilling and Peter Kenny's lab, they took these birch rounds, they surface sterilized some, they autoclaved others, and then they challenged them with a white rot and a brown rot fungus. And there's some really interesting results that I'll run over time if I go into too much. But what they showed is that both the rate and the fate of the quality of the materials, so how much lignin, and stuff remained, were affected. So that leads me to the question of, can we predict decay progression, sort of these distinct pathways of decay, by reconstructing community assembly histories? Probably not, and at least not in this one isolated system, but we can work toward this goal. And so I hope that the work that we'll do here will aid in that effect. So here's the approach that we took. We went to a single forest stand in Denmark. We sampled from a gradient of living trees. And from these, we extracted DNA and then amplified for prokaryotic and fungal organisms. Then we sampled from a gradient of decaying trees, and from here we extracted DNA for fungi, prokaryotes, general eukaryotes to get protists, arthropods, collembola, and nematodes. Now, when I went to the field and I saw the logs and I knew that inside could be something like this, it led me to ask, how do I sample this most appropriately? I want to get a representative sample. And, do you grind the whole log up or what do you do? So from now we use the whole stem of this seedling, but from the rest of the living trees in decay stage 0, we sampled at regular intervals. And then in these other decay stages, we used a more targeted approach where based on the presence of fruit bodies, we would sample from around that area. So in each decay stage, we looked for Fomes fomentarius, which is the most common white rot fungus there. Fomitopsis panicula, which is the most common brown rot fungus there. And then Eutypus spinosa, which is a very present ascomycete that causes a type 2 soft rot. And I want to stress that during this time we took a lot of measures to collect sterile samples, and then also took non-sterile samples so that we could measure these physical chemical parameters. So we used an elemental fingerprinting approach to track trace elements, and then we're looking at like density, pH, moisture content, and then secondary cell wall structural components. Now we used a metabarcoding approach, as I mentioned, and a lot of the thinking here comes from these two papers if it's not cited down here. But the power of this technology is really in multiplexing, where we're putting some identifiers at the end of our amplicons so we can pool samples and then later separate them and tell what they are. Now, this presents a lot of problems, but I took 720 PCR products into 2 tubes that were sequenced on 2 lanes. So some of the potential pitfalls and efforts to mitigate those. As part of the sequencing preparation, we have to worry about tag jumps where something from sample 2 moves over to sample 1 and we misidentify it. So it doesn't prevent this from happening, but downstream we can fix it by using double tagged and dual indexed amplicons. PCR chimeras are a big problem, where this is thought to happen where the elongation is aborted, then that fragment is used as a primer in subsequent cycles, and then we have 2 fragments coming together, which if you can't look at that manually, you think it's a very unique and cool species, and that can happen so many times that it causes major problems. So for that, we're using this Lulu post-clustering curation that takes into account how these chimeras are formed. Primer contamination, so you have to order tag primers. You have options of how you do it. Do not order plates. It's so easy to get them mixed up and you can't go back once you do that. So order them with individual lids, and that's what we did. The workflow is really important. So we use separate pre and post PCR labs, and if we ever worked in a post PCR lab, we didn't go back. So once you have amplified DNA and working in the same area, it's going to contaminate. And then we used a lot of measures to ensure sterility. And another tricky aspect of this is having proper controls. So the negative controls are fairly easy, right? We can use PCR blanks, create negative controls along every step, but the positive controls have not been used much in the past, but now we know that we should include these mock communities. So I included 3 mock communities for fungi, prokaryotes, and insects, and then also to cover the other organisms that were there, 3 known samples from a previous study. And then after all that, you can still have problem with the sequencing, the way that the samples are recognized or sequences are read by the Illumina machine. You get what's called sample bleeding, where something from sample 2 can look like the same color as sample 1, and so then it gets mixed up. So having increased sequence diversity, and if you have low diversity, like you're only going to use fungi, then include this phi-X viral genome to increase that. And then lastly, after getting the data, there are pitfalls in the bioinformatic pipeline, usually to do with the clustering and chimera detection algorithms. And I like to use this as an example. Mick Watson from Edinburgh said a PhD student put 21 microbiota sample through bioinformatic SOPs. With chyme, got over 1,000 OTUs, and with mother, over 500 OTUs. Be afraid, be very afraid. And I thought it was interesting. Somebody from one of these communities responded and said, well, you used the wrong settings for what you had. And I think that really illustrates the point, is that small tweaks in these bioinformatic settings can have huge effects down the line. And pretty much what all of those problems cause is this OTU overinflation, where it looks like we have this massive alpha diversity, and beta diversity could go either way depending on how the samples get mixed up. Okay, finally, onto the analysis. So we're going to use a network analysis approach, and when so every organism that was sampled for, or that we have sequence data for, and every environmental parameter becomes a node in one of these networks, and then they're connected by what we call edges. Now there are also pitfalls with this that have to do with just how sequence data come out, and that's the first dimensionality, is that we typically have many more species than we do samples. They're sparse, so there are a lot of zeros in the data, where most species aren't in most samples. They're compositional, typically, where, as opposed to, let's say, plant pollinator networks, where we're observing a bird go to a flower. Here, we're using abundance, and it's relative abundance. So we could have 10 samples, 10 sequences in 2 samples of the same species. But if we have another 10 here and 0 here, it's going to look like there are a lot more in sample 2 of this species. And these are complex data sets. So with a 5,000 OTU matrix, you have potentially 12.5 million two-way interactions, and we know that there are multitrophic interactions. So these are difficult to infer. And so what you can potentially do, if you have solid data, you can potentially say, oh, this is a mutualistic relationship, and this is commensal, and this is parasitic. But right now, where the technology stands is that you can plug the same data into different technologies, get wildly different results. So we won't do that. But what we can do reliably is look at the topology of this, the structure of the network. And one characteristic that I'm interested in is centrality. In the simplest sense, centrality is the number of links incident upon a node. So this has a high degree centrality compared. But we can extend that and say this closeness centrality is, let me get this right, it's the average length of the shortest path between all nodes. And so here, this has a high closeness centrality. And then another centrality measure is this betweenness centrality, where some species or abiotic factors are acting as bridges between 2 different modules. And these modules are defined as being dense within, having dense connections within, but sparse connections between. Okay, and then down the line, I hope to assess the networks to see how robust they are to external perturbations. So if we looked at a random network, we would expect that most nodes would be averagely linked. But nearly all real networks have this power law distribution where most nodes are lowly linked and a few nodes are very highly linked. And I think the best analogy for this is the internet, where if you imagine Google is going to have a ton of connections, Facebook a ton of connections, but all of our personal web pages don't have that much. And in a random attack, it's thought that the more heterogeneous, these are called heterogeneous networks, the more heterogeneous it is, the more robust it is to a random attack. Of course, target attacks are a different story. But we can measure this heterogeneity, and here they call this a giant complex. And we can see if we do simulate habitat fragmentation, what happens? Does it start to break down? Well, here we still have a pathway, so it's thought that that giant complex isn't broken down yet. But what if we simulate climate change? Then we can see like maybe we just get a total breakdown. We don't have connection through the network, and this can have implications. Maybe the community will be more prone to invasion by an exotic pathogen or extinction events. And then I hope to, when I know what we think are important species in these networks, to see if any of them are already red listed. Maybe we need to enact some extra measures to make sure they're not lost. But also this could be used maybe as a metric for red listing species to see how vulnerable they are to being lost. Okay, so just to summarize, in part one, we said that quarry roots host diverse endophytes. 11 of the dark septate endophytes that we isolated strongly inhibit the Phytophthora. I have 22 that I still need to screen, and in the future, I want to identify the secondary metabolites, look at the difference between asymptomatic, symptomatic communities, and develop a biocontrol strategy. Then in part two, we sampled environmental DNA from a gradient of living and dead wood. We used many field lab and hopefully bioinformatic approaches to produce high quality data, and then plan to use network analysis to predict decay progression, assess robustness, and identify key species or sub-communities that may be important. And with that, I'd like to thank Landcare Research for having me, especially Maj Padamsi and Stan Belgard and Peter Johnson and Ducktool Park, who helped me out a lot. And then the Royal Society New Zealand for their support, and then SMEC in Denmark, and the Danish National Research Foundation for their financial support, and then Jakob Hillman Clausen, Tobias Fruslev, Rasmus Collier, and Bo Dalsgaard. And with that, I'll say, take any questions.